Thank you very much for the invitation and the opportunity to show you part of our work. We are at the Max Planck Institute for Neurobiology of Behavior, but we don't do neurobiology. So what we are mainly interested in is how single cells perform complex computation, and our single cells are outside of the single neuronal cells. What I will mainly show you is work on epithelial cells. But we have seen already today in the talk of Jordi and several others that cells are faced continuously, whether they're being in a complex tissue or in an organ, they're faced with signals that change over time and space. So they continuously have to sense and interpret a complex environment. And they have to do this in real time in order to determine their phenotypic state. And this is very different, I would say, than how computers work. And I would like to argue as well that single cells, they have to balance between opposed features in contrast to the classical computers. So what do I mean by this? If we take a simple example of an immune cell that chases a bacteria, or a cell during embryogenesis or wound healing that has to migrate over a complex tissue, then it's faced with signals which are disrupted, they're noisy, and they change over time and space. So the cell has to account for robustness in its motility in order to bridge these larger distances when the signals are disrupted, but it also has to stay responsive to the changes in the signal. So it has to balance between robustness and plasticity on one hand. But also single cells have to interpret complex time-varying signals, which means that they have to account for specificity in the signaling to establish a specific phenotypic response, but also to be able to generalize signals. So what I mean by this, I would like to show you in one example. This is a work from the lab of Perth a few years ago, where they used PC12 cells, which are neuronal stem cells, and when they are stimulated with epidermal growth factor with specific frequency, they showed that a given frequency of, for example, 3-minute stimulation and 60 minutes interpulse interval, or 3-minute stimulation, 3-minute interpulse intervals, they proliferate. But if you change, you use the same chemical signal, but you just change the frequency to 310 or to 320, these cells will differentiate. So this means that the cells can actually establish specificity in their signaling because they can establish different phenotypic output, even if they see the same growth factor signal, but they also can generalize. So they form classes of the inputs in order to match to a specific phenotypic state. So if we think in forms of, let's say, artificial neural networks, when we would like to construct a network that has specificity, we would think of constructing a modular network, but when we think of generalization, we're going to construct a distributed network. And here we have a case that we have the same network, biochemical network, that is under the epidermal growth factor receptor, which is capable of actually performing both features at the same time. So what we are interested in is how these opposed features, such as robustness, plasticity, but also specificity and generalization, occur in a same biochemical network in single cells. So let me start with the first examples, and we were interested exactly how this robustness and plasticity happens for cells that migrate in dynamic fields of chemical gradients. So if we have signals which are disrupted over time, it is necessary for the cell to have some kind of a memory to be able to bridge with the directional migration, the range when the signal is disrupted. Because you can imagine that if the cell starts with a random walk migration every time the signal is disrupted, it will never be able to reach a long-term distance. But not only that they need to have this memory to bridge when the signal is disrupted, this memory should not stop the cell from being responsive when the signals change in space and time. So we wanted to know how these two features are accounted in a single cell, and for this we used experimental systems of the epidermal growth factor receptors. A few years ago, we did a large experimental study. We tried to understand the basic proteins that interact with EGFR, that control its phosphorylation state or phosphorylation dynamics, and we could reconstruct also the core or the minimal network motif that regulates EGFR phosphorylation dynamics. And we have this great experimental system where we have the receptors fluorescently tagged, we have also the ligand fluorescently tagged, and we have protein tyrosine binding domain, which is fluorescently tagged. So basically we can record simultaneously both the concentration of the receptors, their phosphorylation state, but we can also look at the fraction of ligand-bound receptors, since we also have the ligands which are fluorescently tagged. And we can put these cells into microfluidic devices where we can generate complex gradients, and we can simultaneously measure the polarization of the signaling across the plasma membrane of the receptor, which then generates a polarized state that guides the migration, and look at the output of the migration as well when we have the complex signals. So we did this experiment, and this is one experimental kymograph. On the x-axis, basically, we take the cell, which is now stretched. So we look at the phosphorylation of the receptor across the cell membrane, and each line here represents time points. So we keep the cell in a gradient for 1 hour. This is between these green lines. And then we switch off the gradient, and we follow how the phosphorylation state of the receptor changes. So we can see that the receptor phosphorylation is polarized in the direction of the signal during the presence of the gradient. But we can also see that this phosphorylation is maintained for a transient period of time after we switch off the signal, and then the system resets to a basal phosphorylation state. So this maybe it's better seen when we look at the temporal integration of the signal. So we see that phosphorylation state in green here is marked the duration of the gradient. We see that EGFR phosphorylation is increased. When we switch off the signal, we see that we have transiently maintaining this high phosphorylation state before the system resets, which means that in the phosphorylation state of the receptor, the system keeps a memory from the direction where it has previously seen a signal for a transient period of time. And in this, it encodes the information from the environment it has seen in the previous, in this case, 40 to 50 minutes. This is the average duration of EGFR phosphorylation that we see in single cells. So the cells then, we looked, of course, into the migration response, and the cells translate this transient memory in receptor phosphorylation into a memory of directional migration. So we stimulate the cells with a gradient from the left. The cell starts moving in this direction. When we remove the signal, this is now the red part of the trajectory, we see that the cells continue moving in the direction where the signal was previously. This memory is transient. It is for approximately 40 to 50 minutes on average in single cells. And after they lose this memory, then they will start into a random walk a migration, which is the typical response for epithelial cells in absence of the signal. So this means that cells have a memory in terms of receptor phosphorylation. They keep track of the environment or what has changed in the environment, and use this memory in order to generate robustness in the response when signals would be transient, for example. But then the question was, where does this memory in receptor phosphorylation comes from? So can we understand it from dynamical perspective, and can we understand what kind of features this memory will give to the system? So in general, in dynamical systems, but you probably also know this very much from neuroscience, when we would like to understand the state of a network, we measure several components of this network, and then from this we can construct the collective state of a network in terms of the trajectory, and then with certain dimensionality reduction techniques, which we already heard as well, we can look whether these trajectories lie on some low dimensional space, and this allows us to associate some theoretical concepts, for example, of a line attractor or a ring attractor, and try to understand these information processing features in this language. In our case, however, we did not measure multiple components in the network. We only measure a single one, that is the phosphorylation state of the receptor over time. However, we have tools from dynamical systems theory that we can actually reconstruct even from single observable. We can reconstruct how the collective state of the, in this case, signaling network looks like. So what we have seen from this reconstruction is that when the cell sees a stimulus, it moves from the basal to the polarized activity state, and then when we remove the signal, the system does not reset immediately to the basal activity state, but it gets trapped in this area in phase space here, close to the polarized state. So since we know in details the topology of this network, we can also analyze it from dynamical system perspective. So we know that the basal and the polarized state actually represent steady state of the system, and this memory that we have seen in the polarized receptor phosphorylation, this comes from a so-called metastable ghost state, or so-called attractor ruin. So basically when the polarized state is lost, the system gets transiently trapped in this metastable state, and this is what the cell has as keeps as a memory from where it has been polarized, or in which direction it has been previously polarized, and use this memory in order to generate memory in directional migration. So we thought, okay, since this now memory does not come from a steady state, but it comes from a metastable state, then probably it will be, this will also give the cell flexibility to respond to changing signals. So we constructed an experiment where we first introduced the signal and a dynamic signal from the top side of the microfluidic chamber. So we started with very steep gradients and then go to very shallow gradients. Then we wash out the signal, but then during the period of the memory that the cell has, we introduce a second gradient from the same side. So with this, we mimic how the cell would respond when the signal would be disrupted from the cell from the same side. And then again, we wash out that gradient, and now we reverse the gradient from the opposite side. So if our hypothesis was because the memory, as I mentioned, comes from a metastable state, then the system will be able to integrate all this information and follow the changes in the signal. So we have also done a physical model of the cell in order to mimic how the signaling in the protein activity changes the shape of the cell. So what I show you here is one of these simulations. So we introduce first the signal from the left side. The cell moves in this direction. It has the memory of the previous signal. So when the signal is disrupted, cell actually continues moving in the direction where it has previously seen the signal. But then it is also capable of rapidly changing its direction when we switch the direction of the signal. Again, even from the last signal, the cell has a memory where it has seen the last signal. It continues moving in that direction even when that signal goes away. And when it loses this memory, because it is transient, it starts moving in a random direction. So we did that as well experimentally. As shown on the scheme, we introduced disrupted gradients first from the left and then from the right, and then we see that the cell can integrate and move in the direction of the signals even when they are disrupted, but then they can also very rapidly reverse the direction, move to the new signal, has a memory of the last signal they have seen, and when they lose this memory, they go into a random walk migration. So this is very different. And this type of memory where the system has stores information over time, but it still maintains responsiveness to new signals, is something which in Neuroscience Encyclopedia is a definition for a working memory. So this means that the cells have a form of a molecular working memory that enables them to integrate information that change over time and space. And this is very different than if you consider that the cell would have a permanent memory. In our theoretical consideration, this would be if the system works only with the switching between stable attractors, so being away from these ghost states. And in this case, already the first signal is going to lock the direction in which the cell is moving. So then when the next signal comes or changes localization, that the cell cannot resolve, or at least it cannot resolve it in the simulations in a real physiological relevant time. So it takes a very long time for the cell to be taken out of the stable attractor and to be able to follow next signals. And this is also true if you consider how cells would respond to two signals from two different directions which have different amplitudes. So if the cell works in the case when we would have a working memory, the cell very rapidly resolves what is the signal with the stronger amplitude. It moves in that direction. Again, it's going to show memory of the last position where the strongest signals has been seen, and then switch into a random walk migration, whereas the cell with the permanent memory actually cannot generate a response in distinguishing in which direction it should move. So working memory, or this type of molecular working memory, is also helpful for cells to discriminate when they see simultaneous signals which have different amplitudes. So we wanted to describe more in details why the working memory works in single cells from dynamical systems point of view. And since we work with a very small system, we can also calculate the potential, quasi-potential landscapes that describe the internal state of the system in terms of the number of steady states. And we can also quantify the basin of attraction of these steady states, so we can see how the steady states, if and how they would guide the behavior of the system. So one thing that becomes obvious when we work with transient signals or signals that change over time is that the number and position of the steady states of the system changes. So this means that we cannot apply anymore the classical linear stability analysis, because if the system continuously changes the number of steady states, then we cannot describe the system anymore through the steady states. So we propose that it's necessary when we work with systems that are under influence of time-varying signals, that we have to describe the systems through the trajectories. And one thing that also comes obvious is that when the system has such ghost states, then the trajectory of the system spends substantial amount of time outside of the steady state. So this again argues into the direction that we need to move from the description of the system from the steady state to a description of the trajectories, because this is going to give us a more detailed understanding of what happens in the dynamics of the system. And also these trajectories or transients that give rise to this memory in the systems are not bound. The dynamics is not bound by the steady state, so we don't talk about asymptotic, but rather about non-asymptotic transients in this system. So we have, in order to be able for living systems to be both robust and plastic, the system spends substantial amount of time outside of steady state, and this is what gives them both the robustness, because the ghost states give them transient stability, but also it gives them the plasticity in order to be able to respond to upcoming signals. And we could also construct calculate how these landscapes look like for these ghost states. And they have this characteristic shape of something that looks like a chair. Basically, they are attracting from three sides, and they are repelling from one side, which can explain. And when the system comes to this seat of the chair, it actually has the shape is slightly tilted, so the system would move slowly in this direction. And this is what we have observed as this memory or this ghost state. So basically, the system utilized these ghost states as manifolds of slow points in order to be able to bridge the dynamics in the system and to integrate the signals that come over time. So then we wondered whether if the system or the EGFR network has exactly these manifolds of slow points or ghost states, would this be actually helpful for the cell to be able to generalize the classes of time-varying signals? And can we explain the example I've showed you at the beginning, that if we just change the frequency, the cell will have different phenotypes in terms of proliferation and differentiation? So how we understand this is that depending on the frequency of the signal, for example, if the second pulse comes when the system moves away from this manifold, then the system is going to make several transitions back and forth, so it will have one type of response. But imagine that the second input or the time-varying signal, the next pulse is going to come when the system is in this slow manifold. So basically, the system is just going to move on this slow manifold, which means that the overall dynamics will be integrated in a different way. So we wanted to understand whether these manifolds of slow points can be used to integrate time-varying signals. So we went back exactly to the example of the PC12 cells, because we have there exactly stimulation with EGF. We have our EGFR system where we experimentally determine the length of the memory, and we made a hypothesis that if a system has a working memory in the receptor level coming from such a Go state, which is a metastable, then the system can generate a mapping for class of inputs to a trajectory class, and under trajectory I mean the signaling state of the network, and this will be interpreted as a different phenotypic state. And there will be a unique mapping that always a specific class of signals are going to lead to a specific phenotypic state. So we took the EGFR network from a larger EGFR network from a database that we have modeled. We stimulated this network with the different classes of signals that produce proliferation and differentiation, as in the examples, and we look at the signaling trajectories. So we can see that the classes of signals like 3,3 and 360, they induce proliferation in the experiments. We can see that they have a very characteristic shape of the signaling trajectory, which means that they generate a very similar signaling state of the biochemical network, whereas the classes of signals like 310 and 320 that generate differentiation, they generate a very different trajectory in phase space, so which means they generate very different signaling state of the network, and this is how the cells could possibly interpret the signaling state to different phenotypic outputs. So we also, well, the First Lab also provided experimental data, but we have in the meantime also acquired our own data. So we stimulate PC12 cells with the different signals of EGF, with the different frequencies in microfluidic devices, and we record activity of one of these signaling proteins, ERK, using fluorescent sensors. And again, we can reconstruct the phase space trajectories from single cells from the experimental data, and we see that indeed the signaling trajectories that correspond to proliferations for the very different signal frequencies like 3,3 and 360, they generate a characteristic shape in phase space, meaning they generate a characteristic state of the signaling network, and the state of the signaling network would be different for the class of signals that generate differentiation. So why this is the case? Well, I would like to demonstrate this with a simple example. So we have imagined that we look at how the phase space trajectory is generated when we have the 360 and 310. One is for differentiation, the other one for proliferation. So when the next pulse comes, if the system is, and here is the region where the system has this Go state of the slow manifold, if the pulse comes when the system is in the slow manifold, the next pulse is going to shift the trajectory to a different area in phase space. And if the next pulse comes when the system has gone through this slow manifold, then the system will actually have a very different route in phase space. And this is how these slow manifolds can actually control or generally regulate how the cell, having such a working memory, can integrate very different signals. So to summarize, we have shown both theoretically and experimentally that cells have form of a working memory that comes from Go states, which they use to navigate in continuously changing environment and generate both robustness and plasticity, but also this working memory helps them to map inputs to phenotypic states, and thereby have both the features of specificity and generalization with the same network. And that signals class is determined by the memory length of the receptor. For example, the TRK receptor has a very different length of the memory, and it's going to integrate the signal classes, the frequencies in different classes, so different frequencies are going to generate different phenotypic responses. So we would like also to argue that having very advanced computational capabilities are emergent properties of biochemical networks, and something which I have not discussed, but what actually all this is based on is that all these computational capabilities come about because the systems are organized at criticality, and we think this is a fundamental feature for living systems to have advanced computational capabilities. And with that would like to extend next to the idea whether cells use this type of ghost states for learning. And with that, I would like to thank you and all the members of my group who contributed to this project, as well as our funding sources. Thank you.
Okay, we have time for a few questions. Thank you.
That was really, really cool. I'm just curious, so you were showing these tokens embeddings of the individual cells, and we've actually played with that a little bit ourselves, and you often see like incredible heterogeneity. So if you look at one cell, you get one shape. You look at the next cell, you might get the same shape, you might get a very different shape. And I'm just curious if you played at all with that, and in particular with the decision making that you're talking about, proliferation versus differentiation. Oftentimes cells don't all do the same decision in response to the same signal. The heterogeneity in the population allows for the kind of graded response that Wallace was talking about. So I'm just curious if your sort of slow manifolds give you insight into that kind of thing as well.
In the second part with the PC12 cells, no, we have not done that. We have done that in the first part where we looked at the migration. So the second part, I also have to say this is not published, and it's something that we are trying to finish now. Yeah. In the first part, we have quantified the morphology of the cells, but in a very simple way, just to distinguish between non-polar and polar state of the cell. But where we see the heterogeneity is whether the systems sit at criticality or not. And this is directly related to the concentration of the receptors on the membrane. So there we find, at least for the cells that we analyzed, 80% of the cells were exactly at this, tuned at this criticality region, and they exhibit all this transient memory and so on, but the remaining part of the population would either be pre-activated, which means that it's already on the other side, or was not responsive. But in the case of the differentiation and proliferation, no, that we haven't. We see, for example, from when we reconstruct the signaling, we see there a cell-to-cell variability. But the shape remains the same in the signaling response.
In a Procrustes sense, or what? How do you mean the shape?
If I can go back, shape, I mean of the signaling trajectory.
Just the way it looks to you visually,
yes,
not in a quantitative sense.
I mean, this we quantify as well. So we also quantify Euclidean distance between the different trajectories to look at the similarity of those that belong to a specific class, phenotypic. But this is the, we have only done this type of analysis, I would like to say, for the moment.
That was wonderful. Thank you so much.
Thank you.
Why do you call them ghost states? There seems to be an implication that it's something that was unexpected. And also is in the dynamics. No. Also, just an observation in the psychological literature, if I'm not mistaken, working memory is a whole lot shorter than what's going on up there. I mean, that's short-term memory. That's not working memory. So it's longer than it looks.
Yes. So I would like to first comment on the second question. Well, I think, at least for me, it's very hard to make a distinction between short and working memory. Yeah. But working memory, I understand from the definitions I have read, that's something that is dynamic. That it's not that it stores information over time, but the system remains responsive. Yes. And that's why we call it a working memory, not to the duration. Regarding the first question, the ghost, that is a terminology that comes from dynamical systems, because you have an attractor that has been ruined, but the system still shows a behavior that is similar to the behavior within the attractor. So in the dynamical systems literature, this has been invented as that it's a ghost of the state that has been previously there.
Yes, very nice. So going back to the phase space reconstruction, in this second case, it's very clear that with 3 dimensions you have enough. This is beautiful. But I was going to ask, in the first case, do you need more dimensions maybe? And that's why, because it was not so clear to me, no?
Generally, I mean, of course, we do the, when we do the phase space reconstruction, we look at the embedding dimension and the embedding delay, and we get 3 as using the standard rules for phase-space reconstruction. Here, in these examples, sometimes we get variability, but it goes between 3, 4, and 5 dimensions. But in the previous case, we always got 3 as the embedding dimension. So